Episode 19, Designing the Perfect Trial. Ribavirin in the Long COVID. Welcome to Ribavirin Reminiscence and Lingering Load in Long COVID-19. I'm your host Anushka, a genomics enthusiast who spent years decoding the intricate blueprints of life to understand how our genetic code battles disease. Today, we are stepping into the realm of clinical trial design, a field where science meets strategy, like crafting a map for uncharted territory. Ribavirin holds theoretical promise for long COVID, but anecdotes and lab data must yield to rigorous evidence. So, what would a gold standard clinical trial for ribavirin in long COVID look like? Joining us is Saakshi, a brilliant master's student in microbiology whose passion for viral dynamics and immune responses brings fresh perspectives to this challenge. Saakshi, welcome once again.
Thank you so much, Anushka. I'm thrilled to dive into this critical discussion about designing trials to bring clarity to long COVID treatment.
Designing a trial for long COVID is like navigating a maze with shifting walls. Each patient's symptoms and causes form a unique puzzle. This complexity makes it hard to pinpoint who might benefit from ribavirin. Sakshi, why is trial design especially complex for long COVID?
Long COVID's heterogeneity is the core challenge. Patients present with diverse symptoms such as fatigue, brain fog, or even autonomic dysfunction, and underlying mechanisms including viral persistence, immune dysregulation, or post-viral inflammation. Ribavirin, with its antiviral and immunomodulatory properties, might help some, but not others. For example, a 2020 study on acute COVID-19 found ribavirin did not significantly improve viral clearance or even mortality, but long COVID's chronic nature may require a different approach. The subjective nature of symptoms like fatigue complicates endpoint selection, necessitating both patient reported outcomes and objective biomarkers. Trials must account for this diversity to identify responders and also avoid misleading results.
If we had the resources to design the perfect trial, it would be like crafting a masterpiece. Every detail deliberate and precise. Sakshi, what would your ideal trial for ribavirin in long COVID look like? What type and structure would you choose?
I would propose a randomized, double blind, placebo controlled trial, ideally multi center to ensure diverse participant representation. We would recruit individuals with confirmed prior SARS-CoV-2 infection or symptoms persisting for at least 6 months or no major organ damage and biomarkers indicating viral persistence, example detectable RNA or immune activation, example elevated IL-6. Participants would be randomized to receive ribavirin or placebo for a short course, say 10 to 14 days, in order to balance efficacy and safety given ribavirin side effects. This design minimizes bias, ensures robust data, and aligns with precision medicine principles. While trials for long COVID are emerging, none of them specifically target ribavirin, hence making this design a critical step forward.
Endpoints are the finish line of clinical trials, signaling whether we have succeeded. In long COVID's complex landscape, Choosing them is like selecting the perfect lens to view a multifaceted image. What endpoints would you choose?
We would need a blend of subjective and objective endpoints. The primary endpoint would be improvement in fatigue and cognitive function, measured by validated tools like the Fatigue Severity Scale or NIH PROMIS, which is Patient Reported Outcomes Measurement Information System, or Cognitive Function Index. Secondary endpoints would include inflammatory biomarkers such as IL-6, CRP, or interferon levels to assess immune modulation. Viral persistence, like SARS-CoV-2 RNA in samples such as stool, blood, or even saliva. Talking about quality of life, SF36 scores to capture overall well-being. Going to the functional outcomes, that might include return to work or carrying out the daily activities, and autonomic functions like tilt table tests for dysautonomia. Measurements at baseline, 2 weeks, 6 weeks, and 3 months post treatment would track short and long term effects, ensuring we capture both symptom relief as well as biological changes.
Just as we sequence DNA to understand disease, we need to sequence long COVID patients to identify who might respond to ribavirin. How can biomarkers help stratify the population?
Biomarkers are key to personalizing treatment. We could use PCR testing to detect SARS-CoV-2 RNA in stool, blood, or saliva, suggesting viral persistence. We could even use immune profiling, like identifying T cell exhaustion, high interferon signatures, or elevated cytokines like IL-6. Protein detection could also be done, which includes measuring spike protein fragments in plasma as evidence of antigenic stimulation. Stratifying participants by these markers allows us to analyze subgroups such as those with viral persistence versus immune dysregulation in order to determine who benefits most from ribavirin's antiviral or immunomodulatory effects. This approach, inspired by precision medicine, could reveal why some patients respond while others don't.
Ribavirin is a double-edged sword, capable of targeting viral replication but also risking harm to the host. How would you ensure safety in this trial given its side effects?
Ribavirin's side effects, including hemolytic anemia, which is up to 61% incidence, teratogenicity, and neurotoxicity, require stringent safety measures. We would exclude high risk groups like pregnant individuals or those planning conception, enforcing strict contraception, monitoring closely like regular hemoglobin, liver function, and neuropsychiatric assessments, offering a withdrawal that includes allowing patients to stop if adverse effects emerge, or use a DSMB, an independent data safety monitoring board to oversee safety data. These steps definitely ensure participants' safety while testing ribavirin's potential, especially in a population already burdened by chronic symptoms.
At the trial's end, we are searching for a clear path through long COVID's foggy landscape. What would success look like? What results would justify further trials or even real world use?
Success would mean statistically significant and clinically meaningful improvements in fatigue and cognitive function, particularly in biomarker-defined subgroups like those with viral persistence. A 20 to 30% response rate in such a group would warrant a larger phase 3 trial. Correlated reductions in inflammatory markers, example IL-6 or viral RNA, would strengthen the mechanistic case, suggesting ribavirin addresses underlying drivers. While acute COVID-19 trials showed limited ribavirin efficacy, long COVID's unique pathology might yield different results, hence making these findings pivotal.
If you could send a message in a bottle to funders and regulators, what would it be? What's the one thing you want them to know about this research's importance?
See, long COVID is a global health crisis, leaving millions with debilitating symptoms and no clear treatments. Ribavirin, while not a cure all, offers a window into long COVID's biology, viral persistence, immune dysregulation, and therapeutic timing. We need well funded, rigorous trials to provide evidence based answers, not just hope. Investing in such research is investing in the health of countless individuals awaiting relief.
Sakshi, thank you for painting a vivid picture of what a gold standard trial for ribavirin in long COVID could achieve. Your insights highlight the path to evidence based hope. Next time, we'll explore what comes after ribavirin, the future of broad spectrum antivirals in long COVID. Until then, I'm Anushka. Keep questioning, keep learning, and stay with us.